Hi, everyone. Thank you for the introduction. So my name is Mayra. I'm a postdoc at the Costa Lab at the RWTH Academy University Hospital. And I'm happy to talk to you today about Nichesphere, which is a method to identify disease-specific physical cell-cell interaction and underlying cell communication networks. So spatial organization of cells is crucial to understand diseases. So it can tell us if the tissue is in homeostasis. It can tell us also about developmental stages of tissues, and it can help us as well to describe disease microenvironments, like tumor microenvironments, for example. But to get this kind of insight, a single-cell RNA-seq dissociates tissues, so the spatial information is lost. We need to integrate it with other modalities. So some technologies to analyze spatial organization of cells, like one of the most standardized methods here is Visium 10X, which consists of spotted arrays where each spot contains multiple cells. It has a very good gene coverage. Then we have spatial coordinates for each spot. However, it's a little bit hard to perform in some tissues, for example, the bone. For this, we have other methods, and for example, physically interacting cell sequencing, or PICSIC by its acronym. And this one also covers all genes. And then here we have the advantage that we will get paired single-cell and multi-cell resolution data. And you can do it in any tissue. And as I'm presenting as an example in bone marrow, I will talk to you a little bit more about this technique. So we have interacting cells in the tissue. Then usually we are interested in cells of different kinds interacting. So we will distinguish these kinds of cells. Then we will sort the single cells and the interacting ones or multiplets, and then we will sequence. And then we will get a data set of singlets and one of multiplets. From here, we can do the convolution to figure out the composition of the multiplets, and we can do cell type colocalization. Here are the challenges where I will focus on. And one challenge in the field is to get condition-specific cell-cell interactions. For example, like if we have colocalization probabilities in specific conditions, can we answer questions like which cell types get together in a specific condition? And how are ligand-receptor interactions related to this? So from these ideas, we've started developing Nichesphere. So we do cell type deconvolution to figure out the composition of the multiplets. And then from here, we can get colocalization probabilities of cell types. Then we will transform this data into differential or condition-specific colocalization networks. And transforming this into networks gives us the advantage that we can apply things like community detection or network statistics. Here, with community detection, we will get groups of cells that are interacting in a specific condition. And in order to figure out how these cells are communicating, we can give Nichesphere as input cell-cell communication data. And then we will figure out how the different niches are communicating inside niche as well as between different niches, and through which biological mechanisms in each condition. So how are we getting from the deconvolution to the colocalization networks? So we have different tests. For cases like the PICSIC, where we have one data set per condition, we will ask for each cell type pair if we can find it colocalized more often than expected by chance from a null distribution. So our null model will come from the singlets. So we will take the singlets and make random multiplets from them and check if our observed probability to find the cell type pair is deviated from this random distribution. We will do this with a log odds chi-square test. Then for the communication data, we will take the communication through one specific process. We will filter and just take into account the differentially colocalized cell type pairs. And then we can use tools like Crosstalker, which is a tool developed in the lab, by the way, to get LR communication in each condition. And we will check if the cell type pairs communication scores follow the same distribution or not in the two conditions. And we will test this with rank sums test. Then how are we doing the process-based communication and why? So usually when we have ligand receptor analysis, we use very big databases like CellPhoneDB or Liana, which contain thousands of ligand receptor pairs. And then we get like thousands of significant scores per cell type pair, which makes this hard to interpret. So this is why we are doing the subsetting. We will subset the LR pairs depending on the processes they participate on. You can input like any database that does this. In this case, I will talk to you about a fibrosis example. So as in fibrosis, we get extracellular matrix in places where it should not be. And there's some immune cell recruitment related to this. We are testing for processes related to this. And now I'll talk to you briefly about our case study on bone marrow fibrosis. So if you have a healthy bone marrow, you have the bone, you have the osteocar cells stick to the bone, and there are some other cells in the marrow. There's some signaling, but it's homeostatic signaling. And what happens in a fibrotic bone marrow? We have the bone here, and suddenly some profibrotic signaling starts to appear. And then these guys, the osteocars, they start detaching from the bone and then producing fibers. And then the megakaryocytes also start acting weird, producing fibers. And the questions that we are trying to answer here is who's producing this profibrotic signaling and who's telling these guys to produce the fibers. For this, our collaborators, Helen Glatz, who was before at the Schneider lab, she performed PIC-seq on wild-type and fibrotic mouse models. So from here, we got our data set of singlets, and then the multiplets from which we inferred the cell type, the multiplet composition, and then got the colocalization probability of each cell type pair. Then we applied our test to check which cell type pairs are interacting more often than expected by chance. And using this data, we assembled a colocalization network for the fibrotic model. Here, the nodes are the cells, and the weight of the edges are the scores from the test. Then we applied community detection, and we got these 4 groups, from which we will highlight the fibrotic core as it's interacting more in the disease. So these are the cells that are getting together in disease. And to further interpret, we did network statistics such as betweenness, score, and degree. And this tells us that the SPP1 positive macrophages are interacting with more cells in the disease, and they are also breaching other cell types. Then, to figure out how these groups are communicating and through which processes, we use crosstalker to find communication. We compared the conditions and then checked which process was being used by each group. And as we can see here, we can see that the fibrosis core is using mostly the ECM glycoproteins. And then to find out which cell types are using this mechanism, we got the scores per cell type and built a network, and then did network statistics on it. And here we can see that the osteocars and the SPP1 macrophages are the cells that are interacting with many other cells, and they are also breaching other cells. This tells us that they might have like a crucial role in fibrosis. And then the next question was like through which ligands and receptors. So from here, we went back to the crosstalker data and looked at specific ligand receptor interactions. And as we are interested in the fibrosis core, we looked at this group first. So these are the top interactions from this group. And you can see that they are mostly, it's mostly the osteocar sending SPP1 to other cells. So this looks like the SPP1 ligand is important for fibrosis, which our colleagues validated using knockout models. So they took our model mice, both the wild type and the fibrotic one, and performed an SPP1 knockout, and then measured fibrosis. And as you can see there, in the control, you can see no change because you should not find many fibers there. Fibrotic model, you can find the fibers before the knockout, and after the knockout, you can find significantly less, which confirms the importance of this of the SPP1 in cells as the osteocars. Then we remember that the SPP1 positive the macrophages are also important, and yeah, they are named SPP1 positive macrophages because they also express SPP1. So we proceeded to look at the interactions from them, and they are sending SPP1 to even more cells. These are very sticky cells, and this also tells us that the SPP1 signaling from them might be important for fibrosis. So now our colleagues knocked out SPP1 in the group where the osteomacs belong, and we can see the same. So in the control mice, there were no fibers, and there are no fibers after the knockout. And then we can see a significant decrease in the fibers. So to conclude here, we developed a method to find condition-specific cell-cell interactions and underlying communication networks. Then we transformed the output of the deconvolution into networks, and this allowed us to use graph algorithms to interpret further the data. Then from our case study, we learned about the SPP1 macrophages and the osteocars, which are important cells in fibrosis. And then we also learned about the importance of the SPP1 ligand, which we validated. Then Nichesphere is already public. We have another example on the myocardial infarction atlas. So this is the tutorial you see in my Read the Docs. And we also held a tutorial here with the University of Padova group, the Di Camillo group. And the data from the tutorial is here as well in the link. And finally, I'd like to thank my group, the Costa Lab, so mainly Ivan, and James and Martin for the useful feedback. Then Helene and Rebecca Schneider, who took the experimental part, and then Julia from Padova, who's doing the downstream analysis. By the way, Julia, James, and Ivan, they are also here, so you can talk to them about this. And thank you for your attention.
Many thanks. We have time for questions. I think I missed what kind of data you use to determine what processes are enriching each niche. So you talk about processes that you use to then filter this ligand receptor pair. So if you can explain a bit how you find those processes in the niches.
Oh, okay. So from the ligand receptor based analysis, we get tables of ligand receptor pairs interacting. So in these databases, like the databases I used are Matrisome and Cytosig, and they classify the ligands in different processes. So from there, I subset my ligand receptor communication table, and then I take only those scores and compare them.
Other questions? I will bring you the mic this time.
Thank you. Very interesting talk. A question regarding can you use this when the cells are not near to each other? So not so when you want more autocrine signaling, for example, when you want to look into it.
The method, yes, like here we are using PIC6, so we are taking as colocalized the cells that are in one single multiplet. However, if you have data like Visium or Codex or a slice, you can do the colocalization using radiuses, for example. So you can use the cells in a radius, and then you can look at the interactions there.
Thank you.
I wanted to ask if you have a way to, how you consider batch effect if your two conditions have been measured in different batches.
So yeah, that's a nice question. Like usually we are correcting the data before. So we have the, yeah, in the PIXIC we don't have this problem. And if we have different Visium slices, for example, we can also, yeah, we can take the scores in each for the slices, but we do a different test here. So we can have the colocalization probabilities of the cell types per slice, and then we can take the distribution of those and compare the two conditions, like condition 1 against condition 2. Like for this, you can do this with the same rank sums test that we use for the communication in this case. But yeah, for this we need like replicates. But until now we don't have like another batch correction.
Okay. Yeah.
Very nice talk. I had a question about, so these I guess hub cell types that you identified. Is that associated with more heterogeneity in the gene expression within that cell type, or are they relatively uniform and they are just interacting with a bunch of different cell types? I wonder if you looked at that at all.
Like how we identify the cell types. Like here we are usually using single cell data. So from here we're just doing clustering and then finding the different cell types based on the transcriptome.
I guess my question is the hubs that you identified, like SPP1 macrophages, are this hub cell type? Like you identified some of these cell types that co-localize a lot with a bunch of different cell types, right?
Mm-hmm.
So I was just wondering what the characteristics of those cell types, like for instance, do they exhibit a lot of heterogeneity across their gene expression within that cell type that would be reflective of their ability or the fact that they're interacting with many different cell types? We can talk more offline. That's okay.
Yeah, we are also looking at that. Like we have multiplets, and we also cluster the multiplets. So we also looked at those clusters, and some, like in most of the cases, there is one kind of multiplet overrepresented in one cluster. However, it's not always like the same.
Okay, I think we have to move on.
Thank you.